Alcohol is often seen as both a social companion and a dangerous vice. Yet the question that has echoed for decades is whether drinking actually destroys brain cells. At first glance, it sounds logical. Pure alcohol is powerful enough to kill microbes on a wound and sterilize medical equipment. If it can wipe out germs instantly, wouldn't it also scorch the fragile tissue of your brain? The truth is more complex. Researchers have tested this assumption in controlled studies, and the evidence reveals a story very different from the grim slogans of the past. While alcohol is indeed toxic in high concentrations, the amount a human can realistically consume before death occurs doesn't reach a level that annihilates neurons outright. That's why medical science no longer supports the simplistic phrase that drinking kills brain cells. One of the landmark investigations that shifted this conversation took place in the early 1990s. A team led by Greta Jensen carefully compared brain samples from people with long histories of alcohol dependence to those who had little or no exposure. They literally counted neurons under the microscope, expecting to see a dramatic gap. Surprisingly, the two groups had nearly the same number of neurons, suggesting that even years of heavy consumption doesn't erase brain cells the way many had assumed. That finding has since been replicated in additional work, adding credibility to the conclusion. So, if neurons aren't dropping dead, what is alcohol doing up there? The answer is that alcohol interferes with how cells connect, rather than eliminating them outright. Neurons talk to each other through branching extensions called dendrites, and these tiny structures are sensitive to chemical stress. Alcohol can damage dendrites, limiting the ability of brain cells to communicate effectively. The neurons themselves remain, but their wiring gets tangled and weakened, leading to slower reactions, memory lapses, and coordination issues. The good news is that the brain is an incredibly resilient organ. With rest from alcohol, dendrites can regrow and reconnect, allowing normal signaling to return. Even people who drank excessively for years have been shown to regain much of this lost function once they quit. This doesn't mean alcohol is harmless. Long-term misuse creates problems that extend far beyond temporary signal disruption. One serious disorder is Wernicke-Korsakoff syndrome, a condition linked to severe thiamine deficiency. Alcohol interferes with the body's ability to absorb this vital nutrient, and many individuals with alcohol dependence also neglect their diets. The result is a dangerous shortage of vitamin B1, which neurons desperately need to function. Symptoms can range from confusion and loss of coordination to hallucinations and even coma. While some effects can be reversed if thiamine is replenished early enough, lasting damage often lingers, proving that excessive drinking can indeed cripple the brain indirectly. There's also the question of new brain cell growth. For years, scientists believed adults could never generate fresh neurons. That belief was overturned by the discovery of neurogenesis, the ongoing birth of new cells in certain parts of the brain. Alcohol, however, slows down this process. In nutshell, experiments with animals, high doses reduced the production of new neurons significantly. Encouragingly, once the alcohol was removed, the brain overcompensated, boosting the rate of new cell formation as though it were trying to make up for lost time. Yet, if a person drinks heavily for decades without giving the brain a chance to recover, the slowdown in neurogenesis could still leave permanent effects. Researchers are still debating just how serious that long-term impact might be. It's worth mentioning that alcohol isn't solely destructive. Moderate drinking, especially in older adults, has been correlated with reduced risk of age-related mental decline. For instance, a large study in Italy followed individuals over the age of 65 and compared those who rarely touched alcohol to those who enjoyed it moderately. The group that abstained almost completely showed higher rates of cognitive impairment, while those who drank modestly seemed to maintain sharper minds. The numbers were striking. Nearly a third of lifelong non-drinkers displayed some form of mental decline, compared to under a fifth of moderate drinkers. Scientists suspect that small amounts of alcohol may improve circulation or reduce certain risks associated with dementia, though the precise mechanisms remain under investigation. Of course, this is not an invitation to binge. The protective effect vanishes once drinking becomes excessive. Still, the most widespread myths about alcohol deserve to be debunked. During Prohibition in the early 20th century, wild tales spread about the dangers of alcohol. Some claimed it could cause the body to burst into flames from within because of its flammability. Others swore it would steadily obliterate brain cells with every sip. These exaggerations were powerful propaganda tools, but they don't hold up to scientific scrutiny. Hangovers, for example, are not the result of charred neurons, but primarily dehydration. Alcohol blocks the hormone that helps the body retain water, causing excessive urination. That's why even a night of moderate drinking often leaves people with a dry mouth, pounding head, and drained energy the next morning. The biology behind this is well established and has nothing to do with spontaneous combustion or the wholesale death of neurons. Another piece of hopeful news is the brain's adaptability. For centuries, experts believed that once nerve cells were gone, they were gone forever. Now we know that the adult brain not only continues to produce new cells, but can also reorganize itself in remarkable ways. This plasticity is part of why recovery from alcohol's effects is possible. A damaged brain is not a doomed brain. With proper nutrition, abstinence, and time, much of the harm can be reversed, though not always completely. The capacity for healing, however, is far greater than was once believed. To summarize this first half of the discussion, alcohol does not literally burn away brain cells with each drink, but it can disrupt communication, inhibit new growth, and create deficiencies that indirectly kill neurons. Moderation appears to provide some protection against cognitive decline, while excess leads to a range of neurological and systemic problems. The story isn't as simple as alcohol kills brain cells. The truth is more nuanced, layered with both risks and unexpected benefits, shaped by how much is consumed and over what span of time. While the common phrase about alcohol destroying brain cells has been dismantled by research, the broader picture of alcohol's effect on the body and mind is still far from comforting. What emerges is a portrait of a substance that spares neurons from immediate death, but destabilizes their environment in ways that ripple across thought, memory, and physical health. The difference lies in intensity and consistency of use. A single glass of wine might be harmless or even mildly beneficial, but constant intoxication shifts the balance toward harm, not just in the brain, but throughout nearly every organ system. Consider how communication between neurons falters under heavy drinking. The brain is like a vast city lit up with billions of connections, each one sparking with electrical impulses. Alcohol doesn't bulldoze buildings in this city, but it cuts power to entire neighborhoods. Dendrites lose their ability to pass along signals, so circuits misfire and timing falls apart. Speech becomes slurred, movements stumble, and memory forms unevenly. These lapses demonstrate that the city is still standing, but its infrastructure is overloaded. Fortunately, once alcohol leaves the system, the lights flicker back on, connections strengthen, and stability returns, unless years of abuse have weakened the grid beyond repair. Long-term consequences reach further than these temporary glitches. One of the starkest examples, Wernicke-Korsakoff syndrome, is not caused by alcohol directly poisoning neurons, but by robbing them of the nutrients they require to stay alive. Without thiamine, neurons wither, and brain regions involved in memory and coordination degrade. People with this syndrome may forget entire portions of their lives or invent false memories to fill the gaps. This condition highlights how alcohol exerts indirect devastation, not through fiery destruction of cells, but by creating an environment where they can no longer survive. In that sense, the myth of alcohol killing brain cells contains a shadow of truth, but only through deficiency-driven collapse rather than immediate chemical annihilation. Beyond nutrient absorption, alcohol reshapes brain development in younger individuals. Adolescents who drink heavily risk impairing growth processes that are still unfolding. The teenage brain is still refining its wiring, pruning unnecessary connections and reinforcing essential ones. Introducing alcohol during this stage interrupts the pruning process and can leave permanent marks on decision-making circuits. That's why studies consistently show higher risks of memory issues, impulsive choices, and learning difficulties in those who began drinking early and often. In older adults, by contrast, the same moderate consumption may protect against decline, showing how age, timing, and dose completely transform the outcome. Another fascinating dimension is neurogenesis, the birth of fresh neurons in adulthood. While alcohol does not kill off existing neurons, it certainly suppresses the arrival of new ones. When animals were deprived of alcohol after prolonged exposure, their brains responded with a surge of cell birth, almost as though nature had been waiting to compensate. This rebound never comes, the alcohol never stops, leaving long-term deficits. Whether this suppression translates into permanent memory or mood problems in humans is still debated, but it emphasizes once more that alcohol shapes the brain's vitality not by mass destruction, but by slowing renewal. The conversation cannot be limited to the brain alone, because alcohol's systemic effects bounce back into neurological health. The liver, responsible for detoxifying blood, suffers under heavy drinking, and once scarred, by cirrhosis it fails to filter toxins effectively. Those toxins in turn circulate into the brain, worsening confusion and impairing judgment. The nervous system beyond the skull also feels the impact. Nerve damage in the limbs, a condition known as peripheral neuropathy, leaves individuals with tingling, numbness, or even burning pain. All of this feeds into the central reality that while neurons may not be directly obliterated by alcohol, they are entangled in a larger body, whose deterioration eventually harms them too. Cultural. Myths and moral campaigns once exaggerated alcohol's destructiveness, but they served a purpose. By claiming each drink killed brain cells, reformers created a simple, frightening image that stuck. Even though science has softened that image, the message of caution remains relevant. Excessive drinking destabilizes communication, blocks nutrient absorption, reduces cell growth, and damages other systems that support the brain. It's not a fire that burns neurons down, but a corrosive mist that makes them less effective and more vulnerable. Still, the nuance matters. Understanding that neurons do not vanish instantly after a night of drinking gives people room to hope and heal. The brain can adapt, regrow dendrites, create new neurons, and recover from many forms of damage if given time and care. Recovery programs emphasize nutrition, rest, and abstinence precisely because the organ has this astonishing capacity to rebuild. For someone terrified that years of drinking have left them permanently diminished, the knowledge that the brain retains resilience can be deeply motivating. It shifts the narrative from hopeless destruction to one of repair and renewal. Of course, moderation is the line that divides potential benefit from inevitable harm. Small, regular amounts may promote blood flow and protect against certain forms of cognitive decline. But excess overrides these perks quickly. Drinking patterns matter just as much as total quantity. A steady rhythm of small doses might be tolerated, or even helpful, while irregular binges of massive amounts inflict chaos on the brain's delicate systems. That's why health guidelines stress not just limiting how much alcohol is consumed over time, but also avoiding sharp spikes of intoxication that overwhelm the body in one sweep. As science has advanced, one more revelation has reshaped the conversation. The brain is not static. It isn't a fixed block of tissue that deteriorates without hope. It is dynamic, capable of continual transformation. That fact alone undermines the old fear-driven narrative of alcohol irreversibly torching neurons. Yes, harm exists. Yes, years of abuse leave marks that cannot always be erased. But the potential for renewal remains, and with it, a reason to approach alcohol not with fatalistic dread, but with careful awareness. It is not the inevitable destroyer once imagined, but neither is it a harmless indulgence. In the end, alcohol's story is one of paradox. It can shield the aging mind in moderation, yet unravel memory and coordination in excess. It leaves neurons intact, but silences their conversations. It stifles new growth, then triggers compensatory bursts of renewal. It connects people socially, while isolating them through dependency. To understand alcohol is to accept this duality and recognize that choices shape which side of the paradox emerges. A glass raised in moderation may bring warmth, but bottles emptied without restraint summon consequences that stretch across the body and the mind. The myth of alcohol killing brain cells may be outdated, but the caution behind it carries more truth than many would like to admit.